My dear student, colleagues, and all the viewers who are watching this program live from Facebook page and YouTube channel, I'd like to welcome you all to our International Physics webinar. Good evening to all here in Bangladesh and a very good noon to all those who are watching the program live from UK. Hope you are well and safe from Corona pandemic. Today, I would like to welcome you all to our 113th International Physics webinar. And all you know that we are staying in a Corona pandemic situation, so we cannot continue our normal academic program inside the campus. So we have to start our online program to continue our academic program inside the campus. So I think you have already come to know that our department, Department of Physics, Pabna University of Science and Technology, has started its online program, including the online International Physics webinar. We have successfully completed our 112th International Physics webinar, including 2 Nobel laureates. So we're trying to adjust with this new normal situation. And today, it is a very important day for our department. Today, I'd like to welcome you all to a joint session between the Department of Physics, Pabna University of Science and Technology, and the Department of Physics, Durham University, UK in Physics. And we have with us here today Dr. Professor John Girkin, Director, Center for Advanced Instrumentation, Department of Physics, Durham University, UK. He's connecting with us from UK through online. He has already connected with us, so I'd like to welcome our speaker. So, sir, good evening and good noon to your part. Welcome to our International Physics webinar and welcome to our university in Bangladesh through online. Thanks for accepting our invitation, sir. So, for those who are new, I'd like to, So, for those who are new, I'd like to inform that we have divided our webinar into 3 parts. First of all, I'd like to introduce our speaker with all of you, and then our speaker will deliver his speech, and at the end, we have a discussion time, and in that time, anybody can join with us. I think you have already come to know the title of this today's International Physics webinar, and title is The Physics of Everyday Product, and our speaker is Professor John Girkin, Director, Center for Advanced Instrumentation, Department of Physics, Durham University, UK. And we can see his career and the research of our professor. So Professor Girkin is currently the Professor of Biophysics at Durham University, UK, and the Director of the Center for Advanced Instrumentation. His first degree was in physics from Oxford University, and his PhD in high-resolution atomic spectroscopy and atomic hydrogen. He then worked in industry for 10 years, developing optomechanical product and then clinical instrument for Hiller Limited, including the world's first diode laser for retinal photocoagulation. He then moved back into academic and started Clyde University, Glasgow, UK, where he was one of the leaders in the Institute of Photonics, focusing his research on the application of photonics technology to life science and clinical challenges. He led the development of the physics and life science theme across Scotland before moving to Durham in 2009 to found the Biophysical Science Institute, looking to apply all the expertise of the physical science to help answer challenging life science question. His research has always focused on practical solution to real world challenges, so with a strong focus in the use of photonics and optics. His current research ranges from the development of miniature imaging system for looking inside the lungs through the early detection of dental disease and the development of the novel optical microscopy methods for deep imaging. Since returning to academia, John has published over 100 peer-reviewed publications, and he is currently a deputy editor of Optics Express. He is a fellow of both the Institute of Physics and Optical Society of America, where he was cited his pioneering the use of adaptive optics in optical microscopy. So thanks for all of your patience. Now it's time to go to our speaker. So thanks, sir, for accepting our invitation. So it's your time, sir. You can start your session.
Thank you very much, and thank you very much for that introduction. I'm going to say one comment at the beginning here. I once had some feedback from undergraduate lectures I was doing about 8 years ago, and I actually like to think it's a positive because the feedback came that it said, "I hate this lecturer," and I thought that's not a good start. But then it said, "He makes me think." And if at the end of this, I've made you think, then I've done my job. So what I'm going to talk about is, in essence, some of the physics and things that goes into everyday products. Or if I was going to put it another way, perhaps looking at the world's smallest washing machine. And you'll see how this brings together lots of different aspects of physics to try and understand what's happening. A lot of it from linking into the life sciences. But I thought I should actually start with a little bit about Durham University and where it is. It's the third oldest university in England, and it's focused around Durham Cathedral. And crucially, on the left-hand side here, we have a castle, and part of the university is in this castle, which is about 1,000 years old. In terms of where we are, this is my map of the United Kingdom. And we're up in the northeast near Newcastle. So those of you that are aware of football would know of Newcastle United. So we're about 20 kilometres from Newcastle. Those of you that are interested in cricket, Durham is very close to Chesterley Street, one of the test match grounds in the UK. So we're up in the northeast of England. Its other claims to fame, apart from being a very old university, is also parts of Harry Potter were filmed in different areas of the university as well. So you will have seen the inside of some cathedrals and some bits of the university. This is what it looks like from the air. So the science laboratories are just off the image on the right-hand side, but this is the cathedral, and many of the buildings around there form the old core of the university. So having set the scene, I'm going to talk a little bit about my background and the philosophy that I take to doing physics research or linking physics research. And then my focus is on light, and we'll have a little bit about looked at what can happen to light, and then look at 3 everyday applications of the photonics technology and the physics that I work on, which is looking at dirty washing, shiny glasses, and ice cream. So as you've just heard, my PhD was in atomic spectroscopy, and I then worked in industry for 10 years for a small company. I'll show you a moment about that. And then the sort of underpinning thing in all the research that I've done is often transferring technology from one field to another. So understanding the technology, understanding the challenges or questions in the other field, and then bringing the 2 bits together. Just as, again, a little bit of background, this is the product on the left-hand side was what I designed in 1987 for manipulating light into optical fibers for a company from Photon Control. This has actually been sold as a company several times since then, but the same product that I designed is still in the Thor Labs catalog, one of the large optics catalogs. This was in 2018. It's still there today. And this was the world's first diode laser pumped neodymium YAG laser that I developed, that sort of slightly came out of my PhD thesis a long time ago. At Keeler, as an ophthalmic company, this was how I moved over towards the life sciences. And while there, we developed a puff tonometer for measuring the pressure in your eye, which was the first such handheld instrument, and also laser diode systems for treating, in particular, either retinopathy of prematurity or diabetic-related diseases. What do I do now, apart from this technology I'm going to talk about? One core focus of what I'm looking at is the use and imaging of zebrafish, which you'll be aware are very small, transparent fish, which develop very rapidly. And the advantage that they have for life science research is that they're easy to genetic manipulate, and they are also an excellent model for the human body. So although their heart is only 2 chambers, and a human heart has 4 chambers, 22 out of 24 drugs work in the same way on a human heart. And this is some of the research that we do, but actually only 12 of those drugs would work in the same way on a rabbit or a mouse heart, although one would expect their hearts to be closer to a human. So they're a very interesting model in which to use. And of course, from my perspective, they're optically transparent. The example at the bottom that's shown is of a zebrafish where the heart, which is the green area, is fluorescing due to a natural genetic modification, which means that those areas express a fluorescent protein, so we can study them. I then look at imaging them. So we imaged in this case, and this will come up later, we put a sheet of light in one direction and we look at 90 degrees. So we illuminate and take an optical slice through the sample, and we can do this when the zebrafish is living and intact. It's just anesthetized in a tube, and look at it. For example, in this case, here are some cells. These are neoplastic precancerous cells, and these are the neutrophils in the body, the defense mechanism. And you'll see that we're able to, we will in a moment, when I get it right. There we go. You'll be able to see the neutrophils going inside the cells in real, this is speeded up slightly, but we can watch this happening in real time in vivo in a living animal to try and determine what's happening. And we can also then use some clever optics and some clever electronics to capture images of a beating heart. So again, if I do this, this is a four-dimensional image taken of a beating heart, and we're actually capable of doing this to track where individual cells go. So that gives you an idea of some of the background of the physics and the instruments that I've developed. But now let's look what the approach is. So as I said, it's a holistic approach. It's to try and look at the whole problem. And most real world challenges are not a single subject. You need to bring in some chemistry, some biology, some life science, some engineering, some mathematics to really begin to understand. And the crucial question is, you need to ask what is really required. So if we look at it in terms of perhaps a life science related thing, but it could be in other areas as well. We may start at 12 o'clock in this thing where we image and we observe something and we try and quantify it. And then it may be, as a physicist or a mathematician, we build a model. And then from that model, we try and make a prediction. And then we perhaps go back to the real world and we perturb our system and test it and image it again to try and understand, is our model correct? So have we now understood what it is that we're really trying to look at? And this is the philosophy that I've taken, and we've applied this this sort of perhaps slightly more physical approach to looking at things all the way through. And if we actually look at one of the big challenges is the real world, and I've used here an example in biology, we have to really consider the multi-dimensions. So if we again take the human body, we go from stem cells, individual cells, which have microsecond or less than that for chemical reactions, and they're microns in extent, all the way up to forming clumps, perhaps millimeters of tissue, which then go to form organs and then go to form organisms, where perhaps we're talking over 70 years and perhaps 100 meters if we went for trees. And in fact, if you put this together, we have 27 orders of magnitude. So that's one with 27 noughts after it in terms of the scale that's required in the real world. So that gives you an idea of the complexity. This is life science, but this also applies, as you'll see in a moment, to some other everyday problems. So the philosophy, can we understand what's really required? And then in other words, what is the question we're really trying to answer? And then what's the most suitable method to answer that question? And then to come up with practical solutions, and where possible, use the simplest possible method. So for example, if we want to put it very simply, we choose the right tool for the right job. So one could use an electric hedge trimmer for cutting your toenails, but clearly this is not the right instrument to use. A pair of scissors works much better. So this is all the philosophy. So what we're going to look at in a moment is, can we try and understand what is in essence an everyday problem and use this physics approach to it, to use physics knowledge to try and understand what's really happening. Right, change of subject slightly. What's the complexity of modern day products? So let's just choose what is a very, very simple product. And let's say we have soap. So everyone, I'm assuming, has used soap at some point today. But what does it have to do? This is, this seems like a really simple question. So, for example, it needs to remove dirt. But what dirt? Do we want to remove everything? Clearly, if there was bacteria on there, we may want to do it. But do we want to remove all the bacteria? Some of them may be doing an important job as part of the human biome. So it's an important bit there. So then you go, well, perhaps I've been working on my bicycle or car, so my hands are greasy, so I want to remove all the grease. But do we want to remove all the grease? Because we know that if you really degrease your hands, they become very dry and cracked. So we actually don't want to remove all of it. And in fact, we may even want to put back some natural oils. So this is a really simple product. It's one, as I said, we've all used every day, but there are some really quite challenging questions in there that need to go on. The other thing is we'd actually like it to smell nice as well. So overall, these are all very obvious things. The other thing clearly is, do we want it to be a liquid or a solid? And do we want it to stay as a liquid or stay as a solid? Or do we want it to change at different things? And then the other thing, if you're a commercial company, we want it to have a long life. And we don't want, for example, some liquid soap to separate out into different layers within the things. So this is a really very simple challenge. You can think of some other everyday products, but actually what they're doing has got some deep, complex science behind it. In particular, as you'll see in a moment, some physics. So how can we help? Well, we can develop novel instruments or toys. What are physicists also good at doing? We're good at analyzing complex data, and then we can hopefully develop simple models. These may be different models at different scales. And I think the other thing that hopefully I'm going to illustrate is that the physicists are very good at bringing together different branches of science to try and answer these very often complex questions. So I've given an example of an everyday product, and then how might we go about thinking to analyze this, how this is going to operate. So my background is clearly in light. So what can, just for a moment, take a real step back, what can happen to light? So the most obvious thing that we would see in sort of every day is we see reflection. So light goes in, it hits a surface, metal or a mirror, and the light bounces off. And the crucial bits here, as far as we're concerned as a physicist, is the wavelength. The color of the light doesn't change. We know that the angle in and the angle out are the same. And in perfect reflection, we get no energy change. So this is a very, very simple thing. So keep it, store it in the mind, because we might need to come back to some of this. What also can happen? Well, you can get scattering. So very often when I can use this, and it's a little bit impractical over the internet, I give an example of scattering where you can have a clear bottle of water and you put a drop of milk in there and you see the light then suddenly bounced around in multiple directions. So we have some light going in, it bounces around within the sample and some of it comes out, but some of it will also go back and be scattered back in the same direction or in the same rough direction that the process is taking place. So this is what scattering is. It's now changed. It's going at different angles. But crucially here, the scattering can tell us a lot about the material. So the effect of scattering depends on the size of the particles, the density, how many there are, and the structure. Are they put in a rigid structure in a crystal, which will give a different answer from something that's more random? Why do we get scattering? Because its materials have different refractive indices. This is the speed that light travels through the sample. So different materials have different refractive indices, and this gives rise to scattering. But there's also some important points that we'll want later on. Longer wavelength light is scattered less. Everyone would be aware that the sky is blue, and the reason that the sky is blue is because the small particles in the upper atmosphere bounce blue light around much more than they do the red light, which goes straight through. So longer wavelength is scattered less. And the other thing is that in normal scattering, the wavelength stays exactly the same. So these are all processes we'd be very well aware of. The other thing we can get is light can be absorbed. It's taken in by a particular chemical, and normally it would be absorbed in a short wavelength, so in the blue, let's say, and then a short while later, the light comes back out again. This is something that happens naturally. It occurs in rocks and plants and in animals, and you saw an example earlier of a manipulation where a gene from a jellyfish had been put into a zebrafish to get certain cells to fluoresce. So this is an old process. It's been going on clearly millions of years, and it was first sort of reported in the literature, if that's the right word to use, about 500 years ago, where they looked at fluorescence. They put light at one wavelength from an infusion of wood, and it came out at a different wavelength. Edward Clark looked at it in rocks, and then Brewster in chlorophyll. An example I like to use was William Herschel, who many of you may be aware of, was also very much involved in the development of telescopes, and his sister was also very much involved in the development of telescopes, and was one of the first recorded female astronomers. And he noticed that tonic water fluoresced. The bit that's actually interesting me now is, where is tonic water used? It's often used with gin. And in fact, the gin increases the fluorescence. There's some reasons for this, which is not part of this talk, but it was an interesting comment. And then George Stokes, and you may have heard of the Stokes shift, he talked about this refrangibility of light. And this is an example that was taken in here. There is no gin in this liquid. This is just tonic water with a violet laser diode, so an ultraviolet laser diode, and you can see the light coming out in the blue. So what I'm about to use in these experiments I'm going to show you, they're all very old techniques. So he put quinine in water, as I said, and it's the quinine in the tonic that fluoresces. Stokes used filtered sunlight through a church window, and then he used a wine glass with some wine in it to remove some of the excitation light. Good job the wine didn't fluoresce. And he also talked about a prism. So again, there's an image on the right here. So this is one of the other things that can happen to light. It can be absorbed in a molecule and then comes out at a longer wavelength. Right, so we now know something about light in very simple terms. But then how can we then begin to use it? So one of the techniques that I use is this technique called laser scanning confocal microscopy. So what happens here is we have light coming in from the left-hand side. It goes through a beam splitter, but most of the light passes straight through. It gets focused down by a lens to a focal plane. And light from that is then, there's fluorescence takes place, and the light then comes back through the system, but is this time reflected off my beam splitter. Whoops. And then we go through a lens, through a pinhole, so that light from the focus goes through and is picked up on the detector, but light from a different focus gets rejected by the detector. So we now have a method of actually just taking light from a particular depth within a sample. And we could then take this. So what I imaged just then was clearly just a point. So we could then scan that point in X and Y, and we can build up an image, and then we can change the focus, advance it in Z, and we can then build up a series of images as we go through there to build up now optical slices in 3 dimensions. I talked earlier about an alternative way of doing this in a transparent sample is to put a slice of light and then look at 90 degrees. This is a method which scans each individual point to build up a three-dimensional image. And this is an example of some blood vessels, again taken in this case from a zebrafish. So this is a sort of life science technique that we can use to build up our structure. So I've now given you many of the tools. We've looked at it, we've looked at what can happen to light, and we've also looked at an imaging method. So how can we begin to use these to look at dirty washing? So again, it's not a question I'm going to ask you directly, but I don't know how much of the washing that you actually do, but modern washing machines work at 15 to 20 degrees centigrade. 20 years ago, they would have been operating at probably 40 or 50 degrees centigrade. So a very big difference in temperature. It's better because we're using less energy. But clearly, if we've got some grease on a sample, this is going to create a real challenge, because if it was butter, butter is a liquid at 30 degrees centigrade, but it's a solid. So suddenly our chemicals and what we're doing within a washing machine needs to behave in a completely different way, because we're now trying to remove a solid rather than a liquid. And what actually happens in a washing machine process, so we'll start here where we've got some soil, some fat or something that's on there. So I will come back. We have some fat on there, which we want to break up. So we need to remove it from the material. We then break it into little bits, which can then get flushed away when the washing machine empties. But also we need to do something to make sure that it doesn't get redeposited on the cloth. And also, if there are a few bits that are staining the cloth, we can bleach them. So this is a fairly complicated process that goes on all the time in what one thinks of as being a very simple process. So how does low temperature washing work? We want to break up some fat. We want to use very often an enzyme or a bioactive molecule, and we want to stop it being redeposited. So you go, well, okay, this is just chemistry and perhaps some enzyme biology, but how can we actually understand what's going on if we do this through the physics? So the answer is we can build a miniature washing machine. And we talked a few moments ago about fluorescing labeling. So we can put in some fluorophores, which will label the cloth, our chemicals, and the fat. And we can use the confocal microscope to build up three-dimensional images. But you'll see in a moment, we still have a real physics problem to try and overcome. And so what we actually did here, this is in my laboratory a few years ago now. So this is the confocal microscope. And at the underneath, we've actually built a little washing machine. So water goes round and circulates. The pumps and things are underneath the table. And we we put a red fluorophore in the fat, which loves being in fat and lipophilic. We labeled the cloth using, in fact, fluorophores that are naturally present in washing powders, and we built up three-dimensional images, and we could control the temperature. But there's a problem. And it's the same problem that astronomers have looking at stars from ground-based telescopes. They have to look through the atmosphere, and the atmosphere distorts the image. When we look through, these are from a different sample. This is a soft cheese, but at the top surface, we get little balls of fat. So these fat balls are about 3 microns in diameter. But as we go deeper and deeper in, the image gets more and more blurred because we're having to look through the fat, which distorts our image. So we want to look and see what's happening in our cloth at quite a depth, and we're trying to look at this through fat. So what am I going to choose to try and overcome this? So this is what they do in astronomy, as I highlighted. They have a system. So here's your telescope, and you have an optical element, normally a mirror, that you can change the shape of very rapidly. So if the light is being perturbed by the atmosphere, or in our case, the fat, we can remove that aberration, remove that distortion, and then go on to an image. So we've removed the distortion that the sample is putting in by changing the shape of our optics. We can do this very rapidly. In astronomy, they do it with what's normally with a laser guide star. We can use different methods, use optimization methods in the physics lab, because we can't put a very high power laser through the thing because it would just melt the sample. And this is what happens in astronomy. They take a blurry galaxy and you can then resolve the individual stars within it. In our case, this is from a zebrafish that I talked about earlier. This is what the dorsal fin looks like in a zebrafish before the adaptive optics. This is what it looks like afterwards. So we've made it much clearer. And again, I can scan through this and choose a particular depth. And you can see this one is blurry. Here we can now see all the individual cells again. So we've developed a technique that means we can look through, in this case, a fish, but we can do the same thing to look through our washing. So this is exactly what we did. So this is now a close-up, slightly more of underneath here is the sample, the cloth that we're looking at with a water dipping lens. And this is what it looks like. So we see the fat on the fiber. This fat is in red, and this is the cloth fibers. And we can then do this, and this is an example you'll see in a moment of the fat being removed both by a non-biological washing powder, washing liquid in this case, and a biologically activated one. And you'll see they're going off. This is three-dimensional rendering. Whoops, I will try and stop it. Let me keep it going at some point here. There we go. So you can see here clearly that the biological one in 3 dimensions, we've put it back together with 3 dimensions and time, and clearly that one is removing the material far faster. But we had a problem. What we kept seeing were these big red blobs. And this comes back to, wait a minute, if you see something, what is it that's going on? And we were actually able to see these blobs, and we discovered that actually it was a fat layer, but inside was carbon dioxide. So we asked what chemistry is taking place that produces carbon dioxide, and they said there isn't any. But what we actually discovered is that fat is very good at absorbing carbon dioxide. So what was happening here was that the fat was going in and was going into the material. It was then absorbing some carbon dioxide from the atmosphere. And what we then see here, this is now looking with a transmission image. This is the fluorescence image. And you'll see again, as it goes along, not much happens. And then we could now see these lipid layers with gas inside. They look all red because that's what the fat is doing. But we were able to then pick out these gas bubbles, analyze them, and see what was going on. And clearly this was important because these are very small bubbles. So this is probably about 3 or 4 microns across. But what it was actually doing, which nobody'd seen before, was actually very good at pushing and blowing the fat out from the material. So the company that we worked with here came up with a novel washing powder with slightly different compounds in it, which helps to stimulate the production of gas in there. So stimulates the gas coming out of the fat, which helps the fat to then blow, helps the fat to be removed from the system by blowing it off almost. And we could do a number of different studies. What do we like doing as physicists? Well, we like plotting graphs. So this is a signal level against time for a variety of different things. And you could see these didn't make much difference. But when we put the active compounds in there, the fluorescence, the oil, the fat disappears much more rapidly, which is what we want to try and do. So how has this developed? Well, we then took this information and put it into a computational model. We looked at new formulations of the fat removal, the lipase, and we tested. Suddenly we could do tests using milligrams rather than perhaps 100 grams of compound in a whole washing machine. This meant that the process could be speeded up and done in a much faster manner. And actually, as I said, this turned out to be a product that is now launched by trying to understand the physics in a very everyday product. Another one that we looked at was dishwashing. Dishwashers, glasses, particularly certain plastic items, these are clean, but on the surface, they've developed these water spots. Clearly, this one's in a terrible state, but these water spots appear. And there's glasses with plastic items, and they lose their shine, and it's permanent. It's actually very difficult to then remove this, although it's only on the surface. And it was known why this was taking place, but not how it could be quantified and then studied to try and overcome it. So how can we use some of our physics to try and solve this problem? So we want to try and quantify it. We want to study the buildup. Perhaps from some of the earlier comments I gave about I'm going to use light to try and do this. So what ideas might you have? So one of the things that we started off with, and this was actually a third-year undergraduate project that we gave them, they used a white LED and 3 colored LEDs and a black and white camera. This is a setup, so the camera is over here. This is their light source, which was shining into the glass, and then they were looking at the way the light was scattered. And if you remember, I said that, first of all, different wavelengths of light are scattered in different ways, and also that the amount of scattering that takes place depends on the material and the size of particles and things that are present. So they were able, using this very simple method, to then analyze the data, and they actually came up in this case with a very, very simple way of analyzing it, that they just increased the exposure on the camera and then plotted the intensity of the image over time. For some reason, they much preferred an image that went up rather than down. Doesn't make any difference. So they actually took the black and white image and inverted it, so white became black and black became white, and you then end up with a graph that goes up rather than one that goes down. Doesn't matter. And so then they were able to analyze these, and they looked at the slope. So this is a very white glass, and then this was quite a clear glass which has a low K value. This one, of course, now looks black because they've inverted the image, and it has a very steep curve, so a K value, they called it, of 282. And these glasses are less good. They're not as bad as this, but they're clearly not there. So they could then quantify this and put these all in order. So they've now got a method of looking at glasses just by looking at the way that light scattered. But we wanted to understand what was actually happening. So what could be causing these white bits to form on there? So we came up with the idea that there were cracks, micro cracks in the surface, that when you put the glass in a dishwasher, the dishwasher gets very hot. So what happens when things get hot? They expand, so the little cracks in the glass will get bigger. And but why should it be in some glasses and not in others? And we had to understand how the glasses were made. So cheap glasses, this is a jam jar, but they basically take some glass, they mold it very simply, and then they put gas in it to blow it into its shape. And clearly in doing this, one has a very, very stressed subject because it's been molded and put under large stress. If you use normal, very expensive glass, it's done on a technique called float glass. That is actually left and it's annealed to take the stresses out. So flat glass wouldn't suffer from this, but these curved surfaces or plastics have lots of stress in them. So we look at the micro cracks. So how can we now look at this and try and understand what's happening? So this is not, I should say, how a dishwasher works, but we actually tried an experiment where we did put a camera inside a dishwasher to try and see what was happening. That was not one of my best experiments. There's a lot of water and you couldn't see anything. So can we change something? So the idea was, can we take glass beads, which have a curved surface, hold the glass beads, perhaps using light, and then investigate what's happening, because we clearly don't want our glass beads sitting on a surface because it won't behave in the right way. So we tried to create a micro dishwasher inside a microfluidic channel. So this is a technique using chemistry, very, very small channels, about 100 microns across, where you can flow the 2 bits of chemistry in and they come out. So can we hold our beads inside these channels and to then see what's happening on the surface? And the answer, we can hold the beads, flow different chemistries underneath while we watch what's happening on the surface of the bead. And for those of you that are fans of science fiction, this is from Star Trek. And so clearly in Star Trek, they have this technique where they can pull a whole spaceship along with their tractor beam. Now, I should say we can't hold anything as large as a spaceship, but we can hold small 2-3 micron beads in glass. So we take our glass in here, and these have been around for some time, and it won a Nobel Prize, 2 or 3 years ago for Arthur Ashkin eventually. So the light goes in, and there are different forces. So as the light goes through the bead, the laser's going, in this case, in the vertical direction, it gets bent. And that means that when it gets bent, there's a change in the momentum, which means there's a force which pushes the bead towards the middle. And you can therefore trap your bead in the center focus of your light. It's not very large, but there is sufficient force in there to trap your bead. And to give you an example of how this works, these are 2 micron beads that have been trapped, and they will do a dance for you. So this is a Scottish country dance called Strip the Willow. This was done when I was in Glasgow with someone called Miles Paget, and we trapped the beads and we moved them around and got them to dance as they move back up through here. And so they come to the end. So we can trap these beads and then move them very, very accurately. So how do we do this? We take an infrared laser, we expand the light, and it goes off, and we trap the beads in our sample up here. And we also put in some green light, which also comes off, because we're going to look at a technique called Raman, which is where there is a small change in the wavelength. It's not fluorescence, it's due to the molecular bands vibrating. And we can actually do this with very, very accurate chemistry then to try and understand what's happening to our sample. So we can trap our bead, flow the chemistry across, and observe surface changes due to this Raman effect. And this is what we get for a calcium carbonate. This is a perfect spectra. This is what we get from our sample, and we can monitor particularly this peak here. And over time in our washing machine, you can see dishwasher, it starts to go up and go up. So we can see that there is a buildup of the material, and we're now able to adjust the chemistry so that this buildup no longer happens. And we can do this on a very, very small scale and do repeated samples. So we've developed, using our optics and our physics understanding of light scattering, trapping light, we can then begin to understand an everyday product. But I like to end with something that I think is probably close to everyone's heart, which is ice cream. So in the UK, there is an ice cream called a Magnum, which is very, very nice. These are 3 examples. I apologize. I have given a variation of this talk to some postgraduates, a small group of postgraduates, and we managed to actually give them all an ice cream. I'm very sorry, I can't electronically send you an ice cream, so you'll just have to imagine it. But it's a luxury ice cream, and it has a high fat content. So the ice cream in the middle, encased with the chocolate, has the right crystal structure that with the fat that it's very nice for you to eat. It feels nice on the tongue. And the reason for this is that it's actually made with liquid nitrogen. And so we cool, the ice cream is made, they basically make a chocolate mold, and then the ice cream goes in as a liquid and is then frozen using liquid nitrogen within that material. And the question that you then get asked is, what's the most expensive item? And normally, if I'm doing this in front of an audience, we go round and we ask different people, and people think, well, it must be the nitrogen, or it's the fat, or it's the chocolate. In fact, the single most expensive item is the stick. And you go, well, why would the stick be expensive? And the figures that I had, and I have to put these in English pounds, 10 years ago, the stick cost 1.5 pence, 1.5 pence, but the Magnum ice cream sold for £1.50. So the single most expensive item, ignoring marketing, including transport, was actually the stick. So why spend all the money on a stick? It's actually got to do a lot of very complicated things. Clearly, the ice cream needs to stick to the stick because we don't want the ice cream. It's an expensive ice cream. We don't want it falling off, but we don't want it sticking too much, as you'll see in a moment. So it's got to stick to the stick. But we want it to have a very low specific heat capacity, because the stick is going to get cold. It's going to be cooled by the liquid nitrogen, and we don't want to waste lots of liquid nitrogen cooling the stick. So we want it to have a very low specific heat capacity. The amount of energy needed to cool it or heat it needs to be as low as possible. You also want it to have a low thermal conductivity, because particularly when you're holding the ice cream, you don't want it to come off and it to melt with the stick. So we want it to have a low thermal conductivity, so it doesn't conduct either the heat from the hand out, but also when it's cooling the ice cream, it just cools the stick, the top part of the stick. It doesn't cool the rest of the stick, at least not with liquid nitrogen. And having said the ice cream must stick to the stick, we don't want the ice cream to go too tightly, because we don't want it to act as what's called a nucleation site. We don't want it to be rough, because it's at little points that crystals start to grow. And when crystals start to grow, the ice cream would become crunchy, which is not what we want. So because of these requirements, the stick is made, it's actually made with wood, but it also has an epoxy in there. So they take the wood, grind it up into small particles, and then reattach those particles using a glue and mold it into a very thin but very strong material. So the wood means that it has low thermal conductivity. It's made as small as possible with a low specific heat capacity. Its shape is such that the ice cream does stick to it well. But if you've ever had a Magnum ice cream or Solero or whatever they're called, the stick is actually very smooth, so it doesn't actually let nucleation take place. So you can end up with a very, very smooth stick that has low capacity. So the reasons for the Magnum stick being this expensive are all, in essence, very simple physics questions, physics reasons in the background. So it comes back to the same thing: analyze the problem and think about it using some of these physics techniques that I've talked about, and you can then understand what's going on. So as we come towards the end, hopefully I've given some ideas that you need to use the physics in particular to look at the challenge. In other words, what's the real question that you're looking at? Don't worry initially at what the specific science is. Look at the overall challenge. And then focus on the question. In other words, what is the question that you're really trying to ask? It's to look at the way the fat comes off the cloth. Right, how might we go about doing it? And it happens we used a confocal microscope with some adaptive optics so we could image more deeply. But the crucial question is, we need to watch what's happening in 3 dimensions at high resolution. How do we do that? The other bit is, yes, keep an open mind. Welcome new ideas, brings that things in. And the other thing I've put at the bottom is, why do I do these things? Well, I have fun by constantly learning new things. Keep it quiet, but if I did physics all day long, I might get a little bit bored. But the fact that I'm using my physics, not background and knowledge, in this case, to look at everyday products, but we also do the same thing, as I said, to look at imaging inside the human lung. We were some of the first people to look at what was actually happening in vivo in COVID patients with very high resolution. Again, it was understanding the question, how do we use it? What optical tools can I use? And hopefully at the end of it, I've also given you some ideas that there's some there's interest in even the most mundane products, soap, washing powders, ice cream. Perhaps it's not mundane, but there is some real challenging and questioning physics that will come into it. And hopefully, as I said right at the beginning, if I've made you think, then I think I've done a very good job. So this was a picture. I didn't take this yesterday, but yesterday morning we had a little, not this much, but we had some snow within Durham. It's just raining and cold today, so not as attractive, but this ends. I thank you very much for your attention, and I hope that you found what I've been talking about interesting and gives you some ideas how you can use physics to approach everyday projects, products, and you can then do the same thing to look to apply some of these techniques within the life sciences. Thank you very much.
Thank you, sir, for your wonderful presentation, sir. The main aim of our program is to motivate our student. I think today you have motivated our student in your actually presentation by showing how you can apply your physics in everyday life. So we have some question in inbox. If you allow, we can start our discussion, sir.
Yes, I will show you one other picture. That's what the cathedral can look like in summer from down by the river. So it gives you an idea. Shall I stop sharing my screen?
Yeah, you can, no problem. So the first question, what is optical trapping? Maybe you have already speak about. So the first question, what is optical trapping?
So this is where we use light. So light, as you may be aware, has a pressure associated with it. And the simplest way of thinking about this is if we send our light into the bead, as the light goes in, it's going in initially straight or perhaps at a slight angle, and it then gets bent or shaped by the bead. So that means that my light has changed direction slightly. When any material, anything, in this case it's light, but it could be when it changes direction, there is a force that pushes it in a certain direction, but there's also then a force that goes in exactly the opposite direction. You think of Newton's mechanics. So what's happening here is there's a momentum change in the light. It's changing its direction, therefore it changes its momentum in a particular direction. And then in doing that, if there's a momentum change, that means there has to be a force. And in this case, there is a force that then pushes the bead towards the center. There is also then a scattering force as the light goes in and also then exits the bead. And this scattering force, so the momentum change force is focusing it in the X and Y plane. and then the scattering force is keeping it trapped in the vertical plane. So we use this light that's focused in very tightly, look at the way that the momentum changes of the light, and it goes in. I showed you an example of trapping a glass bead within a liquid. You can actually also track materials. So I have trapped water droplets in the air as well. That's actually much harder because the beads move around more, whereas in water there is some damping due to the water. So this optical technique for trapping things, and if I've got it trapped, I can then move, as you saw, my particle around. I move where the light beam is, and therefore I can then move the particle. And you can do this. You can actually, we're doing some work now on cells from the human lung and looking at the way that neutrophils, the defense mechanisms of the body, come in and attack certain things within these cells from in the human lung. And instead of doing this on a glass substrate where the glass may perturb things, alter things, we can hold the two things in optical traps, bring them together, and actually watch what's going on and measure the forces that are involved. So these are the techniques that we use using optical tweezers.
Thank you, sir. We have another question. So what is the adaptive optics and how adaptive optics help us in detecting black hole or in astronomy?
All right, I can do the astronomy. So the center that I'm now the director of, I put these things on microscopes and small instruments. The other people put them on very, very large telescopes. So the simple way to think about it is that light that comes in, it travels through space, it goes through a vacuum, and there's no problem. When it hits the Earth's atmosphere has a refractive index, and of course it's hotter in small areas than in others. And so instead of getting a nice flat wavefront as it goes through the atmosphere, it gets bent and adjusted. So if we think that our light's coming in and it gets bent by the atmosphere, instead of going onto our detector in the correct shape, it's now going to be bent. So what we can have normally is a mirror, and we put effectively the opposite shape. Because it's a mirror, it's actually the same shape, but think of it as the opposite shape on, so that our light wave goes in, it hits that mirror, and it then comes off being flat. So the aberrations, the distortion that is put in to the light that we're receiving from outside the Earth's atmosphere, we can remove this distortion. The challenge is, what shape do we need to put on our mirror? And to do that, we can either take a single bright star and say that should be a point source, and we can then measure how far it is from being a point source. Or the other way is we can launch, there's a layer of sodium atom in the atmosphere at about 10 kilometers, 100 kilometers, sorry. And we can put make an artificial guide star and look at that, look at the way the light gets distorted from that guide star, and that will then tell us what shape we need to put on the mirror. And in fact, you may be aware there is a telescope. So this is on several large telescopes already, and they actually now produce better images than the Hubble Space Telescope. And there is currently a telescope being built in Chile on the top of a mountain. So 2 years ago, they blew the top off a mountain with explosives, flattened an area, and they are then building a telescope which has a 39-meter mirror. And this telescope will only work because of adaptive optics. Without adaptive optics, you don't need a mirror, don't need a telescope that's more than about 8 meters. If you make it bigger than that, the atmosphere affects the seeing, the quality, significantly. So we can look using this big telescope and see what's happening. How does this affect black holes? Well, one of the methods that's used for detecting black holes is to look at the way that light from distant galaxies get distorted as they come towards us. So you have what's called gravitational lensing. So a black hole, although you may not see it, it absorbs the light, it has a gravitational field. So the gravitation, according to Einstein, bends the light. So we know that if there was a black hole, we might expect the light from a planet to go straight through, or from a star, sorry, to go, or a galaxy to go in one direction, and it gets distorted because of this black hole. Now, we need to therefore measure and determine very accurately where the light is coming from and how much it gets distorted. And if the atmosphere is causing all the distortion, we have no chance of looking for black holes. So producing higher quality images by removing the distortion the atmosphere gives you, and that's how the adaptive optics are helping people to look for black holes.
Thank you, sir, for your answer. So some of our students actually want to study in biophysics. So what he or she need to do for studying in biophysics? So first of all, what she or he need to do?
So as you'll have gathered, I can only speak I speak in two ways. One is, what did I do? I went and my degree and my PhD was in very, very pure physics. But actually, I'm using the techniques and the understanding I got there from within physics to now work collaboratively with life scientists. So the route that I took was, I thought, I'm going to really understand my physics, know my physics field, and then start to work and collaborate with the life scientists that I want to work with. So I did it by understanding the physics, and then thought, actually, it's really quite interesting to try and understand a lot of the biology and the life science thing. So can I then work collaboratively with life scientists? But I still remember that I'm a physicist at core. So the nice thing that means is I can go and I can work with people and ask questions, because my background is different, in a slightly different way, and look at data in a slightly different way. But the good thing is, by working with and collaborating with the biologists, it also means I can ask perhaps what might be viewed as a silly question. It's not a silly question because it's something I don't understand. So can they try and explain it to me? And the same thing works back the other way. They will say, well, how does this happen in terms of light? So I then have to explain the physics to them. So I do all my work. I know that at heart, I am a physicist, but I work closely with experts in the life sciences that want to collaborate, and therefore we put our two sets of expertise together to understand complex problems. So in another area, we're looking at cell signaling between cells. They happen to be blood vessel cells. And to give you an example of the way that we think about them, these are the lining cells of a blood vessel. They're a single layer called endothelial cells, and they do a lot of very important things. And they change their signaling with pressure. But people had done this before by taking a blood vessel, opening it out, and then pulling it. Now, as a physicist, when you pull something, that is not the same force as a cylinder expanding, because in one of them, my cells are being put, they can't, if I pull something, I pull it in one direction, it gets shorter in the other. That can't happen to your blood vessels. Otherwise, every time your heart beats, you keep shrinking like this, which clearly doesn't happen. So how do these cells sense pressure? And we did a series of experiments. We developed a novel imaging system that looked inside intact blood vessels. The biologists developed some novel ways of actually preparing the sample and understanding what was going on. And then we said, well, these cells, they react in different ways. They produce calcium, that is what is used in the signaling. And we worked out that in the cells that were being affected by pressure, because they were being stretched, that means they had to get thinner. And because they got thinner, that changes the way the calcium moves around inside the cell. So we could do a simple model to look how the calcium diffuses within the cells. And we came up with a prediction. And actually, we then did some biological experiments that said the cells do not sense pressure directly, but they get thinner and that affects the way they react to things. So the way that I have approached this is I've understood my physics, and I then will go and speak to them and look at and understand the life science questions, and work collaboratively with people. So I can then work with the best biologists, and they can work with somebody like me, rather than me trying to fully understand the biology. I'm quite happy to admit I don't know very much biology. I have to ask people questions. How does this happen? How do you think this happens? And that's the important part of how I've developed my career in biophysics. It's multidisciplinary. In other words, we bring the disciplines together. We each have our areas of expertise. We're able to explain these to different people rather than me sitting down and learning lots of biology.
Thank you, sir, for your wonderful presentation and nice discussion session, sir. So the main aim of our International Physics webinar, I think I have already told that to motivate and encourage our students. So, and your last motivational speech, I think it will motivate our student very much. And in near future, after the COVID, I will definitely invite you for face-to-face session. And in that time, I will arrange the ice cream for my students. So thanks again for accepting our invitation. So it's my pleasure and honor to host you in our International Physics webinar, sir. So thanks again, and hopefully we'll meet very soon after the COVID session over. So
thank you very much.
Yeah.
Bye. Bye.
Bye.